So our next speaker is Laurel Genzoli, and she is now at the University of Nevada, Reno, but I think this work was conducted while she was doing her dissertation at University of Montana, and I will let her say any other introductory things that she would like to say about herself. She's great. I've worked with her for many years.
Oh, thank you. Awesome. Thanks everyone for hanging into the afternoon. I'm going to talk today about connecting observations of aquatic plants and algae to measurements of river metabolism in an attempt to try to find some other ways that we can monitor attached algae, especially as it relates to nuisance growth conditions. I'm going to end this presentation presenting a little bit of data from these surveys that we've been doing for the last 6 years, with the most recent years of data from the last 2 seasons, just so that I can end with some really current and dam removal relevant results. Okay, so benthic algae blooms are inherently really hard to study. They're super patchy. When you look one direction, you get a whole different scenario on the bottom of the river than when you look to your other direction. So it can require a lot of samples to get a good representation of what's going on in a reach in terms of the algal growth or the algal biomass that's building up. Also, in many rivers, the Klamath included, you can encounter fast and deep water that can make surveying really hard, not only difficult and awkward, but also dangerous or just completely inaccessible at times. And in part because of this, a lot more research has been focused on lake algal blooms than on river blooms. But we get a lot of similar consequences in rivers when we have algae blooms. Low oxygen conditions are common, especially at night. We can have altered food webs and altered habitat. There's cases where toxins are produced in these blooms. And you often get a lot of biomass accumulation, which causes a whole suite of problems. And the Klamath River, as any of you who have spent any time on the Klamath in the summer know, has these really impressive blooms of both rooted aquatic plants and filamentous algae that build up in the river. And that biomass can clog boat ramps. A couple of years ago when I was out on the river, the Forest Service had gone through and literally mowed out the boat ramp so that rafts could access the river. There's a lot of issues with fishing and tribal fishing practices in terms of algae getting caught in nets, getting caught in boat motors, getting caught in the intake of the jet boat motors. So it can impede cultural practices and subsistence practices in the river. And it deters the way that we interact with the river. Very few of us want to go swimming in the river when it looks like that. And so there's a lot of ways that it affects the way we as humans interact with the river and can really limit the way that we interact with the river. There's also TMDLs on the Klamath River for dissolved oxygen, and that's directly associated with excessive growth of algae. We also have TMDLs for nutrients due to their role in promoting these algae blooms. So it's really central to a lot of the water quality concerns on the Klamath River. But despite these really high biomass and consistent blooms, there has not been much monitoring of benthic algae and aquatic plants on the river. Historically, even on the Klamath, where there is one of the most robust water quality monitoring programs that I'm familiar with. And that's because it is hard. It takes a lot of time. It takes a lot of resources to do this. So I started a PhD 6 years ago, in part because I was really interested in tackling this problem. And that's what you do when you go back to grad school, is you spend a whole lot of time doing something that's kind of hard and awkward sometimes. But that started in 2019 as a pre-dam removal survey. And through a ton of support of folks in this room who have either gone out on the river or helped support this project in one way or another, we've been able to continue these surveys for the last 6 years. And in these surveys, we look at the taxa, the cover, and the filament length of rooted aquatic plants and filamentous algae. And we do this at 11 sites or up to 11 sites during the biggest year, usually 10 sites on the river now, from below the former Iron Gate Dam down toward the mouth of the river. And we spend a day at each reach. We float a little section of river and we survey transects, trying to get around 60 points per reach to quantify the growth of algae and aquatic plants on the river. But like I said, it's challenging. You're in the water, you're deep in the water dealing with currents. I'm really interested in trying to understand other ways we might be able to monitor this nuisance growth that might be more sustainable long-term. And one of the methods that I've become really interested in is called ecosystem metabolism. And ecosystem metabolism indicates reach scale photosynthesis and respiration in the river. So it's an average of what's happening throughout a reach of river. And the way we do this is we use the daily change in dissolved oxygen from night to day. At night, you have respiration occurring, and during the, well, sorry, all the time you have respiration occurring, but at night it really draws the dissolved oxygen low. And in the daytime, you have photosynthesis, which is causing oxygen to go much higher. And so because of photosynthesis, which produces oxygen, and respiration that takes the oxygen away, you get this daily oxygen curve. And we use oxygen measurements either from the work I've done, either from the tribally maintained dissolved oxygen probes. In this project, I set out additional probes to kind of go in between some of those sites. And we can model rates of gross primary production, which is essentially photosynthesis, and ecosystem respiration. And that is what is causing that day to night change. And so that's how the basics of this method that I'm using. And this is a method that's been available for a long time, but just in the last decade or two, there's been a lot of innovations, both in sensor technology and statistical methods that allow us to model many, many days of metabolism. And so you can take that raw dissolved oxygen data, which is shown here. You can estimate out gross primary production and respiration, and then start to look at what's happening across time. And this is a study here where they were looking at how did a stream respond to storms, what happened to the rates of production and respiration in that case. But up till now, most of the work trying to understand what drives productivity and respiration is focused a lot on external factors, things like flow and light and nutrients. But we know there's organisms there. They're the ones that are responding, right? It's plants and algae and other organisms in the river that are actually mediating those external controls. And so I wanted to look at both to what extent the metabolism reflected that type and amount of vegetation in the river, and to try to understand how that was potentially controlling this time series of daily metabolism measurements. And so to do this, I estimated ecosystem metabolism using dissolved oxygen data. We did it for about 100 days in the summer of 2019 at the 11 sites that I showed you earlier along the river, using, again, both the tribally collected dissolved oxygen data and some stations that I put in between so that we'd have higher density. Wow, that was not very fuzzy before. Okay, so we standardized the metabolism time series. And when I say standardize, I essentially fit a curve to it to try to be able to more objectively identify peaks in metabolism. So what the maximum rate of gross primary productivity was in the summer, what the fastest rate of green up was, when those important times in the growth cycle occurred. I also looked at variability in the time series and just the overall mean of productivity throughout the season. And we did this for, again, about 100 days at 11 sites. And then I also took the data from those observational surveys where we had about 60 points per reach. We took a mean reach average of the filamentous algae, the rooted macrophytes, and then just the bare rocks. And we converted that cover to estimates of biomass based on field samples. And then we related the biomass of macrophytes specifically to look at how they related to summer metabolism, with the idea that macrophytes, because they're pretty stable and they're growing initially in the spring and then kind of staying there throughout the season, we expected that the macrophytes would have essentially like a time signal or a fingerprint on this metabolic time series. And so that's what I was investigating. Oh no, I lost a bunch of stuff in the conversion to the PDF. So I'm going to tell you what you guys are seeing here. It's going to be really exciting. So overall, we found generally limited evidence that the macrophytes were shaping the summertime series in the gross primary production. You can still see these R-squared values, and they're generally pretty loose correlations between macrophyte biomass and these different statistics of gross primary production. So I had mean gross primary production, the maximum rate, the timing of the maximum rate, the green up rate, the timing of the green up rate, and a couple measures of variability. And they were generally in the direction that I expected in terms of there being more variability in the algal-dominated sites and the macrophyte-dominated sites, and then also having a little bit higher productivity with more macrophytes. But overall, it was pretty weak evidence. And so, at least from this data, from this analysis, I didn't see an obvious fingerprint of macrophytes on gross primary productivity time series. And so there is at least some data that is really important to be in the field and observing, that we're not just going to be able to default necessarily to looking at a time series to understand what's growing. But we did find that total biomass, so that's the cumulative biomass of the filamentous algae, the aquatic plants, and those biofilm layers, related really strongly with gross primary productivity. Again, I had really pretty colored dots here, but there was a positive correlation where total biomass increased nicely with mean primary productivity with an R-squared value of 0.8. And so that the fact that GPP represented total biomass well suggests that metabolism, summers that have high metabolism may also pretty strongly indicate that there's high biomass occurring. And one thing that's really cool about this is that dissolved oxygen data has been collected for a long time on the Klamath River, but not biomass. And so we have this opportunity to go back in time and look at these, use these water quality time series to model these rates of primary productivity and explore these time series and think about how we have summers with no biomass data, but we have this good indicator that might help us understand what biomass was, and then potentially lay the foundation to do some analyses to better understand what was happening. Okay, one of the surprising results for me, because I didn't have really strong hypotheses around how respiration might relate to macrophytes, is that we had these really tight relationships between macrophyte biomass and ecosystem respiration, such that the largest ER fluxes or respiration fluxes occurred where high macrophyte biomass occurred. And it was driven, interestingly, by a shift from pretty positive GPP to ER ratios to more negative later in the season. So essentially, you can see here the light colored points are early in the season. So that's like June. And then this is July, August, and into September here in the dark points. And so we had these autotrophic conditions. So a lot of productivity occurring earlier in the growing season, and then it got more negative later in the growing season, but at these heavy duty macrophyte dominated sites. And that happened much less at these filamentous algal dominated sites. So here you can see productivity was lower overall, respiration was lower overall, but we also didn't get that big swing from autotrophic to heterotrophic conditions occurring between the early and late summer. And there's a couple potential explanations for that. Those macrophytes, there's a ton of biomass there. They might just be decomposing and causing a lot of depletion of oxygen from the water as they're breaking down, but they also provide all this structure. And so there's the potential for the structure of the macrophytes to be trapping this really carbon-rich water as it was coming out of those reservoirs in 2019. And so they might be acting as traps, slowing the water down and causing respiration to occur. One kind of interesting way to wrap this up, or this take-home message from this relationship with ecosystem respiration and macrophytes, is that there were some, at least initial expectations that I'm going to talk a bit more about here in a second, that that macrophytes might decrease after dam removal if we have increased scour with the dam removal, more sediment loads coming down. And if that's the case, then late summer fluxes of ER may also decrease, which would potentially maintain higher rates of dissolved oxygen in the river in late summer and early fall at times that are important for salmon moving through. So there is a really cool potential water quality implication to having these macrophytes fewer or less dense than they were in 2019. And with the last couple of minutes here, I want to shift gears a little bit and dive into the data set that we've collected over the last 6 years to show you all just kind of a first peek at what happened during the last 2 years, where we had these big influxes of sediment, first from the McKinney fire, and then secondly from the sediment evacuation from the dams last year. And hopefully these will help us to try to predict a little bit and understand what we might expect moving forward in terms of changes to the thick macrophyte beds and the thick algae growing on the Klamath River. And so to what extent have our expectations around dam removal been shown in this vegetation data set? Well, initially, I was really expecting at least temporary declines in biomass due to the high sediments, partly because of scour and partly because of decreased light at the bottom of the river. But these expectations were not met as solidly as I had expected. This is a picture of John surveying at Happy Camp with me last summer, where there were just crazy thick macrophytes growing. Macrophytes were overall fewer than the previous 4 years, but they were so much more dense than I expected with this big sediment pulse. This plot here, each point is the mean macrophyte biomass at a site, starting at the site closest to Iron Gate Dam and going downriver as you move to the right. So you can see there's a nice longitudinal decline in macrophytes every year, where they're the thickest below Iron Gate Dam and they decrease as you move downstream. But you can also see that our first survey was in 2019. This purple line here, it was actually relatively low. And that was a year that followed a big winter flood. And they built up year after year after year. We had increased macrophyte biomass coming where we didn't have these big winter floods. And then in 2023, we had much lower macrophyte biomass. That was after that big McKinney Fire sediment pulse. And then in 2024, also low macrophyte biomass following additional sediment pulses from the evacuation of dam removal sediments. But if you look here, these 2 points, they were not affected by the McKinney Fire. That's above the McKinney Fire influx, those 2 sites. And they were also really low. There was also decent flows that winter. So I guess all to say, I'm exploring this just kind of in a storytelling way, saying that the decreased benthic light and the light scour of these really small sediments might have had a really minimal impact on decreasing the macrophytes and filamentous algae in the river. In terms of filamentous algae, we also saw some suppression of filamentous algae, which is shown in that bright green. The macrophytes are plotted below in the blue color. And you can see in these last 2 years, it looks like there was quite a bit less algae growing at these upriver sites. But downriver, where that turbidity was much less, we had a pretty impressive bloom of filamentous algae still. There was still a lot growing in the river despite these really brown, turbid conditions. And so I'm going to end with a pretty boring looking slide here, but to what extent have our expectations around dam removal been shown in this recent evidence? I don't know that there's a whole lot changing in that section below the river simply due to more turbid water. And it's my suspicion at this point that we really are going to need those high winter flows to scour the river, to clean it out, and to let the river start fresh every couple of summers after a big winter flood. But without those winter flows, it seems like these macrophyte beds just build and build and build despite really turbid, really low light conditions of the river. Okay, and with that, I'm happy to take any questions.
Questions? Michael.
Thank you. Just wondering, do those sea shasta worms live on a lot of these macrophytes?
I don't think so. My understanding is that they're attached to the bottom, so they're potentially competing for real estate down on the bottom of the river, where you either have the filamentous algae or the macrophytes growing, with the exception of this like weird thick mat that we still haven't identified. That's like kind of like astroturf. It's about, I don't know, 1/4 inch thick and dense and feels like a little bit of a spongy feel. They grow really thick on that, and Julie can talk to you more about that if you're interested.
Hey, Laurel, great talk as always. I am curious to hear your thoughts about the type of, if you have any sense of like the type of flood event that is required to remove the vegetation. Is it a bed mobilizing flow? Is it a flow that just sort of rips the vegetation out? Like I was just down at Tree of Heaven this morning. It's still macrophyte beds everywhere. Like, what is it going to take this winter to clear the beds? Is it moving the bed or is it simply ripping the roots of the macrophytes out? Because those might be two very different conditions. Do you have
Yeah, I mean,
ideas
that's
on that?
something I'd love to talk more with you and Jenny and others that think more about flows and sediment movement. But what I can say is that these macrophytes can repropagate from the roots. Some species repropagate from roots and seeds. And so I think we'd actually have to start thinking about like, what are the flows needed to completely clear out the seeds, almost thinking about these seeds and these roots as if they were like substrate when we're thinking about scour and sediment mobility. And so I don't know, but I think it does have to be enough to like really clear out some of that material that's really entrained in the sediment. And because these events with the sediment, it's been so small, it's scrubbed, but it hasn't like actually excavated those roots and sent them away and downstream and out of the main channel.
Laurel, I want to say thanks for keeping this data set going because it is really impressive and it is challenging and complicated and you do it every year. So thank you. I saw her diving in the drawdown sediment river this summer. So she's serious. I wanted to ask if the different turnover time for macrophytes versus algae has an impact on the metabolism at sites that are dominated by either one.
Yeah, I totally think they do. And I actually, that's like a core thing. And I took the slide out because I felt like I was going to be over time. But I have some random secret slides here. And this is kind of a lot going on in this figure, but essentially in terrestrial ecosystems, there's this kind of strong feeling that production and vegetation biomass generally relate. You have generally slower turnover times. And so what you're seeing is reflecting to some extent the amount of primary productivity. In an aquatic ecosystem, you can actually have really high rates of productivity with very low biomass due to the super efficient trophic cascade, essentially, where you have the bugs eating the algae faster than it's being produced, and you can have really high productivity that you might see in the GPP time series, but not a lot of biomass buildup. And that's with a very efficient food web. But with a less efficient food web or certain taxa that build up, you can actually have high biomass, but they're not necessarily being super productive. And so that's why this big question even exists in terms of how do these two things relate, because there are these kind of different trajectories a river can be on, based on the food web dynamics and the top-down control.
Any other question? Okay, last one.
Great, thanks. Thanks for the great talk. Just curious if there's any early evidence that the sediment that's been mobilized has ramped up respiration in the river. Yeah,
I mean, we see these big dissolved oxygen sags that are short term. And I think there's no question that, somebody earlier mentioned that there's a chemical oxygen demand occurring, but there's also a very strong biological oxygen demand occurring from the respiration of all that carbon that's coming downstream. And so I would say for sure in the short term, we're working up the metabolism time series for this more recent data, and we haven't done it yet, but we see the super suppressed dissolved oxygen range, which indicates low primary production, but to the extent that there's kind of long-term increased respiration is something that we haven't gotten to yet, but we are working on. In general, in the Klamath River, in the summertime, most of the respiration is super tightly tied to the production. You see this almost perfect relationship where the more production you have, the more respiration, because it's not almost all, but it's very, very tightly autotrophic respiration that's occurring. It's largely cut off from the terrestrial landscape, and just not a lot of respiration occurring outside of that. And I did just really quickly want to say, because I skipped through this, this project has been super collaborative. There's been a ton of support. The Klamath Tribal Water Quality Consortium, folks at Montana have given a lot of support to this. Oregon State has supported it. And there are so many people in this room. Just out of curiosity, raise your hand if you've been out on a survey with me. There's at least 3, 4. All right, there have been dozens of people over the 6 years that have gone out and supported these surveys. So just a ton of appreciation for everybody that's been out on the river and worked on this data set. So thank you guys.
Great, thanks, Laurel.